Welcome to Biotechnology World. Now our topic applications of bioinformatics. Drug discovery and development. 1. Target identification. Finding potential molecular targets for new drugs. 2. Virtual screening. Using computational tools to identify potential drug candidates. 3. Drug design. Designing new molecules based on the structure of target proteins. 4. Pharmacogenomics. Studying how genetic variation affects drug response. Genomics and personalized medicine. 1. Genome sequencing. Sequencing individual genomes to understand genetic makeup. 2. Disease gene identification. Identifying genes associated with diseases. 3. Personalized treatment plans. Developing treatment plans based on individual genetic profiles. 4. Predictive medicine. Predicting disease risk based on genetic information. Proteomics. 1. Protein structure prediction. Predicting the three-dimensional structure of proteins from amino acid sequences. 2. Protein-protein interaction. Studying interactions between protein to understand and cellular processes. 3. Biomarker discovery. Identifying proteins that can serve as biomarkers for diseases. Transcriptomics. 1. Gene expression analysis. Analyzing RNA transcripts to understand and gene expression patterns. 2. RNA sequence data analysis. Using sequencing data to study gene expression and regulation. 3. Alternative splicing. Identifying different splicing variants of genes. Metabolomics. 1. Metabolite profiling. Identifying and quantifying metabolites in biological samples. 2. Pathway analysis. Understanding metabolic pathways and their alterations in diseases. 3. Biomarker discovery. Finding metabolic biomarkers for disease and conditions. System biology. 1. Network modeling. Creating models of biological networks to understand complex interactions. 2. Pathway analysis. Studying pathways to identify key regulatory nodes. 3. Integrative omics. Combining data from genomics, proteomics, and metabolomics for comprehensive analysis. Evolutionary biology. 1. Phylogenetic analysis. Studying evolutionary relationships between organisms. 2. Comparative genomics. Comparing genomes across species to identify evolutionary changes. 3. Molecular evolution. Understanding the mechanisms of genetic evolution at the molecular level. Agriculture and crop improvement. 1. Genetic engineering. Developing genetically modified crops with desirable traits. 2. Marker assisted selection. Using genetic markers to select for beneficial traits. 3. Pathogen resistance. Studying plant pathogen interactions to develop resistant crops. Environmental biotechnology. 1. Microbial community analysis. Studying microbial communities in different environments. 2. Bioremediation. Using microorganisms to clean up environmental pollutants. 3. Biodiversity studies. Analyzing genetic diversity within and between species in ecosystems. Clinical diagnostics. 1. Genetic testing. Identifying genetic mutations associated with the diseases. 2. Infectious disease surveillance. Tracking and analyzing the spread of pathogens. 3. Prenatal screening. Detecting genetic abnormalities in fetus. Forensic science. 1. DNA fingerprinting. Using DNA profiles for identification in legal cases. 2. Paternity testing. Determining biological relationships using genetic markers. 3. Crime scene analysis. Analyzing biological samples from crime scenes. Educational and research tools. 1. Bioinformatics software development. Creating tools for data analysis and visualization. 2. Database management. Curating and managing biological databases. 3. Training and education. Developing curricula and training programs in bioinformatics. Key technologies and methods. 1. Machine learning. Applying AI techniques to predict biological outcomes. 2. Big data analytics. Handling and analyzing large-scale biological datasets. 3. High performance computing. Using advanced computing resources for complex simulations and analysis. Thank you.